Yeah, okay. So let's get started with the second half of today's session. It's my pleasure to welcome Anatoly Kolomeisky from Rice University, and I'll just let him get into it. So thank you very much.
Thank you. Okay. So good morning, good afternoon, and good evening to everyone. Mostly probably good evening. So I know it's the last talk, and this is the end of the difficult day for most people in India. For me, it's only the beginning. It's 6 a.m. So let us start. So I will be talking about stochastic mechanism of cell size regulation in bacteria. As you probably know, cells is the main building blocks of all biological matter. So what I show on the left is this is blood cells, normal blood cells under microscope. If I show on the right, this is skin cells, normal human skin cells under the microscope. We can look at cells in plant world. So this is an onion epidermis cells. These are the cells when you are cleaning the onion, and many of you did it, you're getting tears. That's because you're breaking these cells and some compounds coming out, which makes you cry. So these are, on the right side, these are plant cells under microscope. You can also look at bacteria. This is bacteria, which is probably one of the most famous, one of the most studied one. It's called E. coli bacteria, and this is a different bacteria called Bacillus subtilis here. So when we look at all of these cells, we actually notice that although the different organism and different parts of the organ, different tissues have different sizes and shapes of cells, for specific organism and for specific tissue, they are remarkably similar. They're very, very similar. The distribution of shape and sizes is very, very narrow, and this is very surprising. So this similarity in the shape and size of the cells of the same type is so important that deviation from this uniformity still remains one of the most important marker for cancer. So what I show in this is a normal cell tissue, and these are abnormal architecture and abnormal shapes of the cancerous, tumorous cells. So it is very, very important for, it seems that it's very important for biological systems to have cells of the same type, of the same tissue, to be the same shape or very close shape and very narrow distribution of sizes. So people believe that this cell size uniformity is a consequence of the phenomena which is known as a homeostasis. Homeostasis is a very important principle that that organisms, that living systems follow. It essentially reflects the tendency to maintain constant physiological properties like blood pressure, sugar level, body temperature, levels of some proteins, whatever. And people believe that this cell size uniformity is a part of the homeostasis, and homeostasis is the way for organism to function the most efficiently. So this is a way to keep the most efficient functioning of organism. And so just to remind you, or to give you examples of homeostasis, temperature. So if you look over there, there is a temperature, and this is obviously in American way because Fahrenheit temperature is over there. This is body temperature. We know that temperature of normal person, not sick, should be 36.6 degrees Celsius or less than 100 Fahrenheit. And any deviation from this is a sign of the disease. Again, any deviation from this constant or almost constant value is a sign of a disease. We know that the pressure in normal, healthy human beings, the pressure should be 120/80. It's systolic pressure over diastolic pressure, and any deviation from this is a sign of the disease. So this homeostasis is a kind of a dynamic state of equilibrium, and equilibrium is in a quote because we know that biological systems are never in equilibrium. So equilibrium for them is death. So it's actually steady state. And it describes the most optimal functioning of the organism. Okay, so how can we understand this regulation of cell size? It is clearly it's a result of multiple biochemical and biophysical processes, and specific size of the cell, it's a result of a balance between cell growth is a mass accumulation, so cell will grow in different directions, and cell division. At some point, one of the goals of the cells to produce the same things. Mother cell should produce to daughter cell, which should be genetically identical and continue so on. And so this balance between the growth, mass accumulation, and division, of people call it cell proliferation, this leads to the size, to specific sizes and shapes of cells here. Yes, we understand this. But what a specific molecular mechanism or microscopic mechanism of cell size regulation? Here the things are not so clear. So people were thinking and discussing this issue for a long time, and when looking at mammalian cells, people come out with a concept. It's called cell size checkpoint. So they argue that there are specific biochemical pathway devoted to this regulation of the cell size, devoted to connecting the growth, the pathways for the growth with the pathways for division. And there is some information on these checkpoints have been received, but it's still very limited. Still very limited. The problem at the moment for mammalian cells, we still do not have enough quantitative information on growth and division. So quantitative is exact size, exact times. It's very difficult to do it because it's essentially population measurements, and they are, as you probably know, from other problems in biophysics, population measurements like ensemble measurements frequently hide the details of single cell dynamics. But there is a bacteria. Bacteria is a single cells. And unlike eukaryotes, unlike this mammalian cells that I showed you before, they do not have cell size checkpoints, or at least nothing yet found. And we actually study bacteria for many years and at a very high level, but no cell size checkpoints were found. No specific biochemical pathway that connects the growth and division have been found. So what is the mechanism of cell size control in bacteria? And important things in bacteria that because of the single cell nature, you can do very quantitative experiments. But before going to experiments, let me argue what are the possible mechanism of cell size control in bacteria that people discuss. There are two main mechanisms people discuss. So the one mechanism is called Sizer model or Sizer concept. And the idea here is that if you start with a relatively short cell, small cell, then during the growth, large mass, large volume is added. If you started with a relatively long cell, like over here, then it's a short mass of volume is added, and so on. And if continuing this way, you will come to a stationary state, to some specific average size here. So essentially, the Sizer model argues that it's a regulation mechanism that selectively restricts the growth of large cells and selectively promotes the growth of small cells. The second dominating concept in the field is called adder model or adder concept. And here idea is drastically different. So the idea here is that during every division, on average, cell adds the same volume or the same size is added, the same size, the same size, the same size. And you can show that after several division, your size approaches some stationary value. If you're larger, if you would start from a smaller size, it will also approach some stationary values. So the idea here is that in the adder model, between divisions, cell accumulate on average the same amount of mass. So as I told you, because of the single nature of bacteria, or at least that you can look at them in a single cell level, there is a lot of information you can get. So as I mentioned, the population data for mammalian and yeast cells are difficult to interpret because of the ensemble nature, if we would like to use these statistical mechanics arguments. But for single cell measurements are similar to single molecule measurements. And that allowed researchers to obtain a lot of quantitative information. So one of the biggest advances in this quantitative studies is a development of so-called single cell microfluidic device called mother machine. Okay, it allowed people to it allowed researchers to collect a lot of quantitative information here. So and here let me show you. I hope the video works. This is how this mother machine works. So what you can see, there is a microfluidic channels, and there are many of them, and you can see fluorescently labeled bacteria, and they start to grow, and then there is a flow on top. And when you above some microfluidic channel, the flow moves away, but washes away extra cells here. But you can really do a huge number of measurements simultaneously. You can measure the sizes, you can measure the number of appearing cells, and so on. So as you understand, the mother cell is at the bottom, and then it starts to divide, and the next divide, and so on and so on. So there is a lot of information and experimental information, and this information allowed people to distinguish between two theoretical concepts, adder or sizer. And experiments which I show over there clearly show that adder principle, that between divisions on average cells add the same amount, the same volume. And here the data comes from two types of bacteria, for E. coli and Bacillus subtilis. And what you show for different nutrient conditions, this is time, and this is added size. As you can see, added size at different nutrient conditions is not the same, but for given nutrient conditions, it is the same, and it's both for E. coli and for B. subtilis. And you can see that E. coli, like human, like sugar, that when there is a lot of sugar, glucose, you really grow quite well. That's what you can see here. So and you can put all the data, and they collapse on a one plot. So on average, you added the same at given nutrient conditions, you added the same size or the same volume. So it's clearly adder principle. So what is the current theoretical views, how it might happen? After multiple studies, people come out with this argument that there are two main principles. So the one principle is called balance biosynthesis. So essentially the idea in a simple word is that they believe that the proteins or precursors responsible for growth and division are created much faster than any time scales for growth and separation. So you can see the proteins responsible for growth or division accumulate much faster than any time scales for division or for growth. Yeah, that seems to be very reasonable, because we know that the time scales of cell division is minutes, hours, while the time scale for producing proteins is in milliseconds. Yes, but the second problem, the second concept, the second principle that people are proposing, in my opinion, is more questionable. So the second idea is that you need to have a threshold accumulation of division initiators or precursors to a fixed number. In other words, the division will not happen until you collect, let's say, 100 proteins responsible for division. 99 division will not happen, but 100, and it will happen. And this is very problematic. It's very problematic for the following reason. So there is a specific biochemical and biophysical processes should be responsible for the appearance of this threshold, but none have been identified in experiments. There was exhaustive search, and still continues, and still they couldn't figure out why there is exactly this threshold, or it is coming up. What is the point of it? So the idea of threshold is phenomenologically convenient, but practically, or from the microscopic point of view, is really useless and not productive. So we come out with a different hypothesis how it might work here. So our hypothesis is very simple. We argue that the cell size regulation in bacteria, at least in bacteria, is governed by several stochastic processes, at least two in the simplest version of our model. It's a cell division and cell growth. They are coupled together, and these two stochastic processes coupled together lead that effectively leads to the appearance of some like a threshold. What is important that this assumption of stochasticity eliminates the need of using the threshold. and as I mentioned, which is the weakest point of current theoretical approaches. So there is no need to have threshold. The threshold comes naturally or appears effectively as a result of coupling of two stochastic processes, or two or more. So let me try to explain the model. How does it work? The model is very simple. So imagine that we essentially assume a one-dimensional description of cell growth. Why? Because as I already showed you before, let me go back to bacteria picture, so maybe you will see. Yes, so look, the bacteria mostly has a rod-like shape, and they mostly grow along one direction. So you can really, as a first approximation, you can really take one-dimensional description. So we have a one-dimensional discrete stochastic model, and we associate some variable, parameter n variable, with the size of the cell. And this corresponds to the number of proteins responsible for the growth or for division. For example, we know that for division of E. coli bacteria, you need to have this FTSZ proteins. Maybe you can associate n with the number of these FTSZ proteins, for example. Then the next assumption in our model that these proteins responsible for cell growth and cell division are formed much faster than the growth and division rates. And this is reasonable for typical conditions when nutrients are sufficiently available, as I argued. So then essentially, we always have a given number of proteins for the growth and division, and the ratio between these proteins for growth and division are always fixed here, and it's always proportional to this variable n. And this is consistent with experimental findings of this balanced biosynthesis, as I discussed a few minutes ago. Good. So this is a very simple model. So in this model, so imagine that I have size n, right? Then only two processes can happen, only two stochastic processes. The cell of size n can grow with the rate n lambda. So the rate is proportional to size. The rate is proportional to number of the proteins available for the growth, or it can divide with the rate nk. Okay, so the rates are proportional to the size, because size, at the given size, there is a fixed number of proteins responsible for growth and some fixed number of proteins responsible for division here. So this growth and division viewed as kind of a chemical reaction. And as I mentioned before, this is important because it allows us to naturally introduce stochasticity in the system and eliminate the need for assuming any thresholds. So what is important that according to this model, the division can take place for the cells of any size, any size. In reality, you would only see division in some very narrow range of sizes, and the reason is when your length is very small, the division rate is very small. When your size is very large, the division rate is very large, but it means that you will never see cells of such size, and there is a balance which will lead to specific range of cell sizes where division can happen. But I will explain this more in the moment. Good. So let us start to analyze this very simple model. So if we have the number of proteins responsible for growth and division is very high, we can use a simple mean field arguments to calculate, to evaluate the dynamics of cell size growth. And you can write this very simple equation, very similar to chemical kinetic equations. So if you look at the evolution of the average length, this is like chemical kinetic equation. It has two terms. So this terms on the left is the first term is very easy to understand, right? This is just a growth. The average length grows proportionally to the average length with the rate constant lambda. Here it's a shortening. It's a negative term, and let me try to explain where this term comes from. So first of all, Kn, this is the rate of division. Kn rate of division. The coefficient 2 in front comes from the fact that every division you're creating two, from mother cell you're creating two daughter cells. And then you multiply by this. That's because you eliminating length n over 2. From original mother cell length, and you eliminating n over 2. So this is a very simple equation, similar to chemical kinetic equations. You can solve this equation easily at all times. You can essentially evaluate the evolution of average cell size, and if you want to do it in a dimensionless way, this is the result here. And what is important that this predicts that there is a stationary average length size, and importantly, it's a balance between growth and division. This is a ratio of growth rate constant over division rate constant. Clearly, what I was telling you, this is a balance between growth and division. So let us try to apply this model to, there is a real data, there is real experimental data on E. coli bacteria and B. subtilis, and let me play this movie, and you can see exactly how this, by the way, measurements are done, and you can see that that if you follow the average length, the length decreases exponentially if it was larger than ever the stationary one, but if it was shorter one, it increases until it reaches stationary value. So the solid lines here are theoretical prediction without any fitting, and the reason without any fitting, because here everything is dimensionless. There is a dimensionless approach of cell size to stationary. There is dimensionless, everything is dimensionless quantity, and the growth rate we take from these experiments, these people measured this growth rate independently. So as you can see, a very simple model works reasonably well. Now, so there is observation, so several observations. There is a very good, excellent agreement with experimental data on these two types of bacteria, and again, without fitting parameters. And what is also interesting, another interesting observation, so bacteria can reach homeostasis very quickly, essentially after 4-5 division if original length deviated more than 50%. Essentially, so you can see it from over there. Okay, you can look at this model in more detail, doing a little bit more precise analysis. If you consider that the system already reached a stationary state, and then you can do more precise analysis without mean field assumption, as was done in the previous slides here. And here you understand that every time when you have a cell size n, it has two possibilities. It can grow with rate n lambda, or it can divide with rate n k. So then you can calculate the probability to divide at the cell length m, and you can see that the probability of division is independent of the cell length. It's the same probability of division every time. So this is an important result, that in our stochastic model, the probability of division is always constant and independent of the cell length. But again, the probability of division doesn't tell you where you will see this division. So using this result, you can calculate the quantity, which is a probability of dividing after adding exactly n units to the cell. And again, this is independent of the original length, and this is a function, and the physical meaning of this function is very simple. There are n times where no division, and on the n+1, there was division's probability p. No division happened with probability 1 minus p n times. And you can plot this function, and this is this function when growth is fast. This is this function when division is fast, and clearly when the growth is fast, you can reach to further lengths. When the division is fast, you cannot go far away because division is fast and only some relatively short length cells will be observed. So physically, this is a reasonable and consistent result here. You can also, using this, I call it random walk approach, kind of random walk approach, you can calculate average added length. And it can be explicitly calculated. All you need to do sum over all possible nqn, and when you do it, you get amazingly get the same result as before. The average length will be ratio over growth rate constant over division rate constant. So essentially, we have a couple important observations. So first of all, we see that independently of the initial size, the same length is added between divisions. So this is the length added between divisions on average. This is the length added between divisions, and this agrees with this experimentally supported added principle. So you can see our very simple stochastic model consistent with added principle, consistent with experimental observation here. And what is important is average edit length is equal to average cell length. We understand why, right? So if you edit length should be exactly the same as the average length. So when you reached twice of this, it divides and you come back to the same cycle and you're staying in a stationary state. Now, the advantage of our theoretical approach, because it's so simple, you can calculate everything analytically, and we can calculate more. And for example, due to stochasticity, as you understand, there is a distribution of edit length. So if I would, I already mentioned to you, okay, so let me maybe for a moment show this experimental data. So let me show experimental data that I showed you. Yes, this one. So look, this is a distribution of edit lengths, you see, for different bacteria. So it's not always the same. It's not delta function. There is some distribution of edit lengths. We can calculate it in our stochastic model. We can estimate it. We can estimate the distribution. So essentially, we can estimate the weights of this distribution. It actually shows a very simple result. And we can calculate what people call normalized variance, coefficient of variance, CV, of the edit cell length. And our theory predicts that the faster the growth, the narrower the distribution. And eventually it approaches the value of 1. So what is also important about theoretical approach, we can not only discuss the distribution of sizes and dynamics of size. We can also discuss the dynamics of cell size regulation. We can discuss the distribution of division times. How long does it take on average for cell to divide, and what will be the distribution of these times, division times? And this can be done with the method of first passage probabilities. We can explicitly calculate this distribution of division times and its moment, and we can collect a lot of information there. So without going into much mathematical details, let me only show this at the beginning. So imagine that you have a cell of size n0, and let us assume that division happens exactly after adding the size L, the one that I calculated before. Then I can define the probability function fmt. It's a probability of dissociating exactly at the length n0 plus L for the first time at time t, if at time 0 the system started at length m. And the temporal evolution of these probabilities are governed by backward master equations, which you can solve. So I will not show you the mathematics. We can solve them. They're not very difficult. And we can calculate a lot of properties. For example, this is the properties, the probability that you can add exactly length L. And this is the result of this first passage calculation. And it's exactly the same function as I mentioned before. So this is a probability. It's independent what was initial length. This is a probability of adding exactly the length L. You can also calculate mean interdivision times after adding exactly the length L. And again, this is exact result here. And you can compare with experimental data. So because of this experiments that people plotted rescaled or dimensionless value, it's very convenient for us. So this red is data, red symbols are data for E. coli bacteria, and blue line is our theoretical prediction. So first of all, we predict that the mean interdivision times decrease with a newborn length. And this is a great experimental observation. And the question comes, why? It's actually very easy to explain in our theoretical model. And let me go back for the model. So our model argues that on average, the same length is added before the division. So if you start for larger length, then it takes faster. It takes less time to reach the point when of adding exactly length L, because each rate n lambda nk is larger. If you start at small lengths, it takes longer because dynamics is slower here. Growth rate and division rates are slow. So this is the argument here. This is because the larger cell size, the length L can be reached faster. And it's naturally comes from our theoretical model, and it agrees with experimental observations here. So we can also calculate any moments of this interdivision times, distribution of the interdivision times, and our predictions, we try to use the value which are very close to realistic one. And our prediction that, look at the scales here, that the noise may be slowly decreasing, but almost independent of the newborn length. So we are predicting that this noise probably will be almost constant, maybe slightly decreasing. And this trend is consistent with experimental observation. Again, everything is analytical, can be calculated and evaluated. Okay, so let us try to discuss the advantages of our theoretical approach. So first of all, it's a very simple physical model. It's a simple stochastic picture of cell size regulation. The model can be solved analytically for both distributions and dynamics of cell size regulation. And this is very important because you can test the model and you can understand a microscopic picture of underlying phenomena. Because we're using this stochastic idea that there are several stochastic processes, we don't need the idea of thresholds. So the thresholds is effective result of this coupling of several stochastic processes is something like a threshold. But specifically, we don't need to have thresholds and we don't need to find biochemical networks or pathways responsible for the threshold formation. And we found a very good agreement with a single cell experimental data for various bacteria. Very simple. So our view of cell size regulation, I'll repeat it already several times. So this is our view, which might be, of course, not correct. The view is that this almost deterministic cell size regulation is due to coupling of several stochastic processes, at least two, cell growth and cell division. And the idea is, why do you see a very narrow range of sizes? That's because if you have a very short cell sizes, short lengths, division rates are too small and cells grow forward. But if you have a very long length, the division rates are too high, and essentially cells with such lengths cannot exist because they will be already divided. The division rate is too fast. And only in some range of the sizes, you have a balance between growth and division, and that leads to a relatively narrow distribution of sizes observed in real world. Good. Now, let us be very critical also, as you understand, all models are wrong. So this model is obviously also somewhere wrong. So what is the problem with this approach? So we simplified extremely complex set of chemical and physical processes. We lumped them up in a two stochastic processes, cell growth and cell division, just to illustrate that this is a very complex biological phenomena, which we try to very naively describe as a single stochastic step. This is a growth, as you can see. This is pictures from a textbook. There is a multiple processes taking place, and we try to simplify them. This is naive to expect that this such simplification would completely describe the cell growth. We also, although I said the agreement with experimental data is very good, it's not excellent. There are some deviations from experimental observations. So for example, when we calculate the distribution of cell sizes, we predict that it should be close to 1, this coefficient of variance, while experimentally distributions are even more narrow. They 0.2, 0.3, this coefficient of variance, normalized variance. So we are thinking about this, how to improve it. One of the ways to do it is obviously to do the following things here. So to assume that this single step is not a single step, but there is some intermediate in the growth and intermediate in the division, and this should probably improve this prediction here. This observation might also tell us that there is how many rate limiting steps in cell growth and cell division. We're working on this problem. We also we also for example, we predict much smaller noise for interdivision times. That's what we predict, while experimentally the noise is larger. That's easier to explain, and the reason is that in my calculations that I presented here, I assume that cells adding exactly the length L. But as I argued, as I showed you, sometimes you can add slightly smaller lengths, sometimes you can add slightly larger lengths, and you can calculate using our method. You just have to add several calculations like this, and then the noise obviously is expected to increase and be close to experimental data here. Now, of course, I use a one-dimensional picture for complex three-dimensional system. Although I argued that bacteria look like a rod-like and mostly grow in one dimension, we still know that they have still a complex shape. You can see the complex shapes over there. So you need to have a better, more realistic 3D growth description. And all my calculations were done for specific good nutrient conditions. So when the good nutrients, so the proteins responsible for growth and division are quickly created, and the rate limiting step in the whole dynamics is just division or growth, but never a synthesis of the specific precursors here. But there are situations when there are poor nutrients, and it will be important to test the model to extend this approach to poor nutrients, as well as to the systems where there are dynamic fluctuations in nutrients or in protein precursors. There are recent experimental data where people were able to artificially stimulate fluctuations in nutrients and fluctuations in the proteins level, and it will be interesting to test our theoretical model, our theoretical approach using these data. So I know it's a long day for most of you, so we decided not to make it very long. So this is conclusion. So we developed a new theoretical framework to explain cell size regulation in bacteria. The idea, maybe it's a wrong idea, but the idea is based on the hypothesis that it's actually pure stochastic mechanism, which when you properly couple cell growth and cell division, you can create, you can come out with a narrow distribution of sizes and almost like a threshold-like behavior. So because of this, our theory eliminates the need of using the concept of thresholds that experimentally not supported, at least not supported yet. And the advantage of our approach is that we have exact analytical solutions, which provides a very clear picture, which can be tested. There is a clear things, clear numbers, and there is also a way to extend the theory in several directions here. We also found a very good agreement with available experimental data. Not perfect, but we do know, as I explained it before, where deviations might come from, and it will be important to extend the theory in these directions here. And so there is one more important thing that I'm thinking. So this was for bacteria, the reason we studied bacteria, because there was a significant amount of experimental information, and because you can really look at single cell data. The question comes, how important this stochastic mechanism might be for more complex mammalian and yeast cells, where there is a multiple processes can take. I believe that there is a stochasticity is also might be important, or at least relevant in what is going in more complex cells, but probably it will need to wait until more data, more clear quantitative data comes from these more complex systems. Good. And I would like to acknowledge that this work was done mostly by my talented postdoc, Dr. Hamid Teimouri, and by visiting student from India who spent 3 months in our lab last summer, Rupsha Mukherjee. And this is financial support, and I will be happy to answer the questions. Thank you.
Thank you very much. We already have a question from Debashish.
Hi, Anatoly, very nice talk, and really amazing that such a simple model can account for so many experimental facts. Now, if I understand correctly, ultimately it is the postulate that you have lambda k and lambda n, right?
Yes.
Yeah. So now, is there, how does the cell adjust the synthesis according to its size? That must be the justification, right?
Right. So when you, let's go one more moment. If you remember, I had a cartoon which probably could explain things better. Yes, here, look. So when division happens, right? Mm-hmm. What is matter for cell number of these FTSZ cells over there? So the reaction or process proportional to the number of these important precursors over there.
No, I understand. My question is that in order to match that situation, the cell must synthesize FTSZ
Yes.
according to the size, right? So it must be sort of tuning up its synthesis according to the size. How does it tune this?
No, I think this is you creating some amount. I think suppose you created 50 molecules, right, of FTSZ. Okay, of course there is more complexity because there is additional mechanism try to put them closer to place where they have to divide. But according to my model, then will be some rate of division proportional to 50. Still will not be large enough. But then only when you reach, let's say, 100, then division will happen. Most probably will happen. So it's a stochastic processes. You know, it can happen even for small amount of FTSZ in principle, rarely, very rarely. But the majority of events will be in this range where growth is compensated by the division. This is a picture which is a little bit fuzzy, I understand, but that's a picture.
So that means the tuning of the synthesis is not required, but according to the synthesis, the division probability gets tuned up, right?
Right. If you remember when I showed experimental data for so this is experimental data for different nutrient conditions, right? And these different nutrient conditions, essentially different growth rate or different division rate, or different, in other words, different synthesis rate of this. But still the size added is the same. So as you understand, because the edit length is not the same here versus here. So which means the growth rate or division rates are not the same in these two conditions. So you don't need to tune a synthesis rate. If for these given conditions, on average, you will get this more or less narrow distribution because of the balance of growth and division. Okay. It might be wrong, but the model is very simple, and you can test it, you know, and you can test it experimentally. So I'm not arguing that this is correct.
Yeah. Since it is accounting for so many facts, so there must be some truth in it. So that's why, yeah. So next question would be, as you said correctly, there's some deviations here and there. So why they are occurring, that's fine. Okay. Thank you.
Thank you.
So I'll ask a question while people are gathering their thoughts. You kept mentioning that you need at least 2 stochastic processes, if not more. But have you thought more about what form those other stochastic processes would take and how they would affect your predictions?
So probably the growth itself consists of several steps, and each of them is a stochastic. It's like chemical reaction, and division itself also doesn't happen immediately, as I showed you. So these are, if you take each of them as a stochastic process and write them properly, that would probably is a way to do it, and that would lead probably to almost more deterministic pictures than I showed up. Okay. So there are several of them. I put it as a one stochastic, but clearly there is a many stochastic intermediate steps. That's what my point.
So you had the division and the synthesis ethers like Poisson processes, essentially.
That's right.
Does adding the additional steps equivalent to just making, say, division and synthesis a continuous time random walk where the weighting distributions may be more highly peaked?
Yeah, something like this. Exactly, something like this. If you splitting this big stochastic steps in a sequence of several of them, in my opinion, would only create a more deterministic picture similar to what was observed. But again, you have to check it. We have to check it.
Yeah.
So if I would have only one stochastic process, I would not. That would be no but coupling two, as you can see, coupling two which work kind of orthogonal to each other already creates this picture which is reasonable.
I see. So any more questions?
Somebody raise a hand.
Hi. Yeah. Hi. Can you hear me?
Yes.
Yeah, so very nice talk. So I was just wondering whether we can apply some kinetic Monte Carlo kind of model in this problem, where this lambda and k will be the rates of a forward step and a backward step. I mean, what will be the problems and how it will, I mean, whether that will be able to predict the total length of the bacteria.
Right, but so this is essentially what is done, but it's exact calculation. You don't need Monte Carlo. Here is the exact calculation. Lambda and k are rates. I calculate exactly. I can calculate exactly everything, and we did Monte Carlo simulations just for students to learn, and of course it agrees because it's the exact solution. But in this case. And that's one of the advantages of the model. It's very simple. It might be wrong somewhere, but it's so simple. You can clearly go and see what is the physics of it. Why you do this and this? Okay.
No, I was wondering. Because I was wondering because there may be some times whether it may or may not divide, or like the probability may not be exact. So
that's right.
So wondering.
Yeah. So the world of cells is very complex. You know, there are cells which divide but do not grow. This is fertilized eggs in embryo. They divide but never grow. There is also cells which grow but never divide. It's some nerve cells, neurons. So the world of cells is very complex, and of course you cannot describe everything with one thing. And for mammalian and yeast there are more complications. Bacteria is simpler in some sense. So I have more hope that this works for bacteria, and that's what I said. It's only for bacteria, at least at this moment.
Okay, thank you.
Okay. One last question, anybody? No, it's late or early.
Both. Both.
Both. Yeah. Okay. I don't see any hand raises. All right. Well, I'll thank Anatoly again and Ambarish as well for their talks today.
So Anatoly, thank you very much for very nice talk. And Jordan, thanks for chairing the session.
Welcome.
Okay, so Anatoly, have a nice day. It's just the beginning of the day, so have a nice day.
Yes, thank you. Have a good night.
Yeah, thank you.
Bye-bye.
Bye.